Hi, everybody. I'm Dr. Suzanne McDonald. I have the honor to be the RCI Science Chair. Welcome back, everybody, to another year of exciting science programming. We hope you had a good summer and are keeping well in these still weird and uncertain times. Before we begin, we'd like to acknowledge that the land we're meeting on virtually is the traditional territory of many nations, including the Mississaugas of the Credit, the Anishinaabe, the Chippewa, the Haudenosaunee, and the Wendat peoples, and is now home to many diverse First Nations, Inuit, and Métis peoples. We also acknowledge that Toronto is covered by Treaty 13 with the Mississaugas of the Credit. Today, this meeting place is still the home to many Indigenous peoples from across Turtle Island, and we are grateful to have the opportunity to work on this land. We know that you may not be in Toronto, and we may do have viewers from across Canada and beyond. So we suggest that you research the history of your land using the website and app native-land.ca. And we also know that talk is cheap, but actions speak louder. So we encourage everybody to work for positive change. So today we have a very good panel. I can hardly wait, honestly. It's a timely panel, and it is moderated by Dr. Amanda Very, our resident fungi expert. So we're going to have an introduction to fungi by Amanda, followed by 3 short presentations by our experts, followed by moderated Q&A and audience questions from you. You can submit your questions and comments in the chat box, or you can do it via Twitter at RCI Science or email to information@rciscience.ca. And just a reminder to be polite and respectful. We do have moderators monitoring the chat box, and we will take action as appropriate. Without further ado, it is my pleasure to introduce our moderator, Dr. Amanda Very. Amanda is a microbiologist who fell in love with microbes during her PhD at the University of Toronto, and who doesn't really, where she studied all the tricks fungi use to cause disease in humans. She's fortunate to work at the University Health Network, where she builds and runs programs to support trainees and scientists, including outreach events to inspire curiosity and support the next generation of scientists. Take it away, Amanda.
Thank you so much for that great introduction. And I'm so excited to bring you this amazing panel of mycologists today, which are people who study fungi. And I just want to start off with a brief idea of what are the basics about fungi. So we're going to learn about fungi 101. And I wanted to start with this picture of a mushroom that I found on a walk through my neighborhood recently, just as a reminder that fall is a fantastic time to stop while you're walking and look for these mushrooms. And you don't have to be in a forest or a place like Algonquin to find them. You can look in any area that there's grass or soil or trees and really see if you can find some of these beautiful fungi. So when I say fungi, many of you might immediately think of this quintessential fungi, which is this mushroom that's red with white spots. And this is an example of a macrofungi, which is a fungi you can see with your eyes and who has this big structure. What's really cool about this picture is I actually drew the picture of the mushroom with a microfungi, which is a small fungi that you can't see with your eye. And in this case, I used yeast, which is baker's yeast. And I just love this idea of using agar art, which anybody who knows me knows I love a ton. But also it gives us this idea of the diversity already, where you have these large fungi and these small fungi, and they're all related and they're all fun. When we think about fungi, we need to think about where do they belong in the tree of life. So we know that all living things are characterized into bacteria, archaea, or eukarya. And so we know that fungi actually are eukaryotes. And this is because they have many of the similar structures like nuclei and subcompartments. And we also know that fungi are more closely related to animals than they are plants. And this is interesting because at first fungi were thought to be plants, and many of the first mycologists actually worked in ecology departments because of that. And this relationship between fungi and animals and humans will become really important for Joshua's talk later on today. So overall, when we look at the family of fungi, we have 8 phyla, which are shown in the mushroom on the left in blue, as well as we have some examples of different kinds of fungi you may have heard of. Some of the general characteristics of all fungi are that they're chemoheterotrophs, which means they require carbon as a source from their environment. They often propagate with spores, and spores of the tiny little kind of puffs of things that come out of the mushrooms that help them spread and grow more. And they also have cell walls, and these cell walls often have chitin. Now, what's really cool about fungi is the vast diversity of them. And I'm going to highlight just a little bit, and first being the diversity of size. So as mentioned in the intro, fungi are actually the largest organism on the earth, which may seem super surprising. There's this humongous fungus called Armillaria fungus, and it has these mushrooms above ground as well as this mycelial network underground. And estimates suggest that this fungi is over 2,500 years old and weighs over 400 tons, and that's larger than 3 blue whales. On the other end, we also have fungi that are among the smallest eukaryotes on the Earth, and this is Microsporidia. So these are unicellular fungi that are between 1 and 4 micrometers, and that's 1,000 times smaller than a millimeter. So not only are they smaller than what you could see, and they're even smaller than those yeast or baker yeast that are also impossible to see on their own. Now, when we talk about diversity of fungi, we can also talk about their function. And of course, with an event called the good, the bad, and the ugly, I wanted to highlight a little bit of each of those. So first, we need fungi to live on this earth, and without fungi, it's estimated that the earth would be inhabitable. And some of the reasons for that are because of things like penicillium, which is a kind of fungus that makes penicillin. And penicillin was accidentally discovered in 1928 by Sir Andrew Fleming. And this penicillin and related antibiotics have saved countless lives and enabled life-saving surgeries because before people used to die when they had surgeries of these bacterial infections. So these fungi are really saving us through these antibiotics. Another example is that fungi in the soil recycle nutrients from dead and decaying organisms. And this is really essential because we have a limited number of nutrients on Earth. And so these fungi are really responsible for bringing those back into cycle. Now, of course, we have to talk about the bad. And crops destroying fungi have been a threat to humanity for centuries. So currently, right now, fungi are threatening our supply of rice, bananas, coffee, and recently I just read about how they're even destroying our pumpkin supplies this Halloween. And so this is really serious because so much of our population relies on these and other crops for food sources. It's not only crops that have to worry, fungi are also threatening many global ecosystems by wiping out populations of amphibians, bats, and much more. And lastly, when we talk about the ugly, there are definitely ugly fungi out there. And one example is the zombie fungus. And if you've heard any of my talks at the Science of Scary, you'll already know this. And if you haven't, you can review those talks. There are these species of fungi that infect and control insects in order to increase the spread of their fungal spores, which I think is pretty ugly. But one of my favorite things about fungi is that even though we know about all this diversity in their function, in their size, and how they look, there's still so much more to be discovered. So it is estimated that between 2.2 and 3.8 million species of fungi are on Earth, but over 93% have not been discovered yet. And so as we discover more about these fungi, we'll really get to see even more about how important fungi are for our life on Earth and beyond. And with that, let's pass it over to the other speakers so that we can learn more about these elements of fungi. So I'm so excited to first be able to introduce to you Dr. Xu Jin Peng. And I apologize, I've tried my best to practice that. You can also call him JP Xu. And JP obtained his BSc specializing in agronomy from Jingzhou Agricultural University, an MSc specializing in agricultural microbiology from Nanjing Agricultural University, and a PhD specializing in fungal genetics from the University of Toronto. After 3.5 years of postdoctoral research training specialized in population biology of human fungal pathogens at Duke University, JP joined the Department of Biology at McMaster University as a tenure-track assistant professor in 2000 and was promoted to full professor in 2012. His current research examines the genetics and population biology of several groups of fungi, including wild mushrooms, plant fungal pathogens, and human and animal fungal pathogens. So thank you very much. I'm excited to learn from you, JP.
Thank you. Thank you, Amanda, for the introduction. And thank you for RCI for organizing this event. I also want to welcome everybody to this forum. It's very heartwarming to have so many people who are interested in fungi. Quite often when I'm teaching over here at the MAC, I'm only interacting with the people in my lab who are interested in fungi, plus a few other stranglers. So I'm tasked with introducing some basic information about mushrooms. So I have here mushrooms good, mushrooms bad, and the bad ones, from my mind, are also the ugly ones. How do I go forward with this? Can I please forward? Okay. So as Amanda pointed out, there are estimated 2.8 to 3.5 million species, but over the last 10 years or so, many different estimates have been made about the number of fungal species in the world. And that range from 700,000 to over 5 million. And these estimates differ because of the different assumptions and also differ because of the species concepts that are used by different people. So out of those estimated species, about 150,000 have been named and described in the literature. So among those, about 10,000 are fleshy mushrooms. Those are the ones that can form floating bodies that we can see from a naked eye, and there's a lot of tissue that we could touch and do whatever we want with them. And about 140,000 are microscopic. That includes many of the molds and the yeasts. So out of the 10,000 that are fresh mushrooms, about 50% of them are too tough to chew and are not really digestible. And another quarter, 25%, they're edible, but they don't really have much flavor and don't really have much cooking or edible quality. And then we have about 44% that are very tasty and nutritious. And those are the ones that typically go out and collect at this time of the year, for example. Now, it depends on where you live, the different mushroom picking seasons. And then we have about 20% that can cause irritations to our digestive system. They usually would not cause death, but cause irritations. And those may differ among different people, just like each of us are susceptible to different things in allergens and other things. It's the same for many of the mushrooms, that some of them can cause severe problems to some people, but may not cause any problem to others. However, about 1% of the total, that's about 100 species, they are highly poisonous, regardless of where you come from, what your background, what your age, what your gender. Now, out of the 140,000 microscopic fungi, about 20,000 are plant fungal pathogens. So here, I'm just showing a few pictures. Amanda already mentioned a couple. These are some of those really deadly fungal diseases that impact farmers and forests. The first one is Puccinia triticea that causes leaf rust. That again, each year causes billions of dollars of loss. The corn smut, caused by the fungus Eustilago matus. Over here, you have the rice blast, caused by the fungus Mechanipothi griesii. And we have here the chestnut blight that basically has wiped out the chestnut trees in eastern North America. And this one is Ophiostoma fungus that causes the Dutch elm disease. Dutch elm trees used to line up the North American streets in the 60s and 70s, and now they're practically all gone. So each of those caused damages in the billions of dollars. For the wheat, corn, and rice, as well as other crop fungal diseases, the estimated damage is about several billions, hundreds of billions of dollars each year to farmers. Now for mushrooms, they can be classified into many different categories based on their phylogenetic affiliation or based on their ecological niche or substrate. So here's a brief classification based on their their lifestyle. So they are classified into broad categories of saprophytes, animal-associated mushrooms, and plant-associated, like mycorrhizal mushrooms. The examples of saprophytic mushrooms, those are the ones that grow on dead organic matter, include the button mushroom. Those are the ones that we typically buy in the supermarkets. They're shown over here. This is what they look like in the mushroom farm. They also include shiitake, oyster, etc. And this group of mushrooms are typically the easiest one to domesticate because of the substrate requirement is pretty well known. The second group are the animal-associated. Some of those examples that people may encounter are in the health food stores, the cordyceps. And I think Amanda alluded to this group as ugly, in that they can control some of the insect behavior. But some of those animal-associated also is a food source for animals. For example, the termitomyces, shown over here on the right. This is one of the species and is associated with termite mounds. And the termites use the mycelium of this fungi as food, as a food source. And when they form fruiting body, we pick them as food for ourselves. And they're yummy, very yummy. The third group is also the largest group of macroscopic fungi, I believe, are very similar to some of the saprophytes. But they play very essential roles for plant nutrition, plant defense against pathogens, and those form symbiotic relationships with plant roots. And the example over here on the right is a russula, very bright red, and are treasured in some of the cultures, indigenous cultures. So I'm going to show a few examples that are really choice mushrooms for pretty much all over the world, and are relatively easy to identify and pick, and are safe to eat as well. So the first example I brought up over here is the golden chanterelle. And they have this very typical coloration and structure. Recently, we went to Newfoundland, my wife and I, and my in-laws. And in Newfoundland, we had a feast around the North Point in Grossmont National Park, where we harvested quite a lot of the chanterelle. And you can see we had a feast, this being a big pot. So this is a relatively easy to identify and choice mushroom for food. The second one, which is also very common in the Northern Hemisphere, is kimbolete, Boletus adulis species complex. And they can be very big. It's called a kimbolete, not for good reason. They're very big, as you can see on the right. A few pieces could fill up the whole frying pan. And again, they're very delicious. Another common one in the pine forest is this mushroom matsutake. And this is found across the Northern Hemisphere, Tricholoma matsutake species complex. They're very highly prized in Japan, in the Japanese culture, which can be sold up to thousands of dollars per kilogram of fresh fruiting body. Basically, about a dozen of this mushroom can be sold that much. But typically it's about $400 to $500 US per kilogram. As you can see over here, especially common in the fall season. And for this mushroom, we have it closely related species in North America. In Eastern North America, it's called Magnivelari. In Western North America, it's called Tricholoma marianum. And in Mexico, there's a Mesoamericanum. And the wild Matsutake collected from different parts of the world are priced differently. And that potentially causes counterfeiting. And we have identified evidence of counterfeiting, geographic counterfeiting of this mushroom. And in the fall, like has been alluded to, it's a very good season for mushroom hunting. And this is one of the common mushrooms that we see around Hamilton. This is the chicken of the woods, different from hen of the woods, which is not as bright colored, but as delicious. So this is my lab from a couple of years ago when we went to South Coots, just close to McMaster campus, harvested these mushrooms from one single log, and we still have quite a lot left over. And each of this is enough for making a couple of dishes for several meals. And of course, this fall, I have seen quite a few giant puffballs. This is one in one of our neighbors' backyard. Now, this one is a bit old. When it's fresh, relatively fresh, it's a very, very solid, round ball that if you harvest and cook it, it could be several meals worth of delicious food. So those are the good ones, based on taste buds. But the mushrooms can also cause deadly consequences. So throughout history, there's been report of mushroom poisoning. Shown here is a couple of prominent examples that have been inferred based on historical text, including the Buddha likely being killed by mushroom poisoning over 2,500 years ago, the death cap Amanita phalloides causing the death of multiple prominent figures in the European history. And there are many species in the genus Amanita that are very deadly. So shown here are 3 of the most common ones distributed in different parts of the world. On the left over here is one that's most common in Europe, Amanita pantherina. In the middle over here is the most common one in southern Ontario around the Great Lakes, Amanita vesperigera, called the destroying angel. And on the right over here, Amanita excitialis. This was only described about 10 years ago, but has been linked with about 100 deaths over that time span, and it's still reported to cause deaths this year. And this one is primarily found in, as I mentioned, Southeast Asia and Southern China. So those Amanita fungi are typically relatively easy to identify because of their unique structure, the bulbous at the bottom, a whale, and the unique cap, identifiable cap structure. But there are many other poisonous mushrooms that are not easily identifiable based on morphological features. And shown here are some of those that can be easily confused with some of the non-poisonous ones. So we have the Russula subnigricans, Garreria, Salsiceps, and the Chlorophyllum over here at the bottom. They are easily confused with some of the non-poisonous and in some of the edible ones. So these mushrooms, we know that they are poisonous. They can cause death. But there's still some of them that we don't know. There's still quite a large number of mushrooms that have not been described, and some of them might be poisonous. So this is my last slide. This one shows a little white mushroom. Up until about 10 years ago, it's not known in the literature at all. It's not described. But Over the last several decades, there's been a report of sudden unexplained death syndrome in southwestern China. And those people die because suddenly, and most of them are very healthy young people, had no prior disease symptoms. They just suddenly dropped dead. And it primarily happens in the summer. So there was a lot of investigative work going in there. And finally, investigators suspected that this mushroom, this little white mushroom, might be the cause. And later on, it was found that it contained toxins that can cause cardiac arrest. Okay, I'm going to stop here, as I have shown that mushrooms are beautiful to look at. Some of them are delicious to eat, but some of them are very deadly. And if you're not sure, just enjoy looking at them, but not eating them.
Perfect. I think that is such a wonderful take home. Those are some really famous deaths that I didn't know of that were caused by fungi. So very important to keep in mind. Thank you so much, JP, for that wonderful presentation. And just a reminder, many of you have already submitted questions, but if you have any questions based off of JP's talk, please feel free to send them in. So next, we are lucky to have a presentation from Joshua Sharma, who is a PhD student from Trinidad and Tobago, who is currently studying the fungal pathogen Candida albicans at the University of Guelph. His current research looks at compounds that can enhance the activity of known drugs or that can disarm the pathogen of virulence mechanisms that make them harmful to the human host. He's also developing novel CRISPR variants to study the functional genomics of a drug-resistant Candida albicans isolate. Outside of the lab, Joshua is a co-founder of CGEM, a Canadian-wide initiative connecting research groups tasked with using synthetic biology to solve real-world problems. His aim is to give students access to the tools they need to conduct research independently and communicate the research on a wider scale to the public. Now, unfortunately, Joshua wasn't able to join us today as planned, but luckily he provided us with a recording so that we still get to learn from him. So with that, we'll learn from Joshua about pathogens and how fungi can be pathogens.
Hi, everyone. My name is Joshua Sharma. I'm a PhD student at the University of Guelph, and I'm here today at RCIS Sciences, the Fungi, Good, Bad, and the Ugly event, to talk to you about fungal pathogens and really taking a look at the more microbial and bad nature of fungi. And even though I'm not with you here in person today, I'm super excited that I still got a chance to send in a video with this presentation. And if you have any questions or comments that may follow, feel free to send me an email, follow me on Twitter. I'll be here and available to take and answer questions. Now, let's get started. Historically, fungi tend to really be thought of as an agricultural threat. We had farming conditions whereby, we have one species like wheat or rice that are planted for acres, and there was and continues to be a significant vulnerability with that. When one plant gets infected by a pathogen, it quickly spreads across the field, and it results in endangering the entire harvest. Now, we've learned these lessons in instances where the fungal like Oomycete, Phytophthora infestans actually decimated potato crops in the 1840s, leading to what we call the Great Irish Potato Famine. These lessons we really continue to learn today when we deal with fungi that can infect and destroy important crops like wheat and rice. We have fungi like Magnaporthe and Fusarium species that can actually infect these crops. And once inside of the plant, they secrete toxins that kill the surrounding tissue. It's these fungal outbreaks of food crops that result in the loss of their viability. And it really reduces our global capacity to feed people by 8.5%. That means that there's approximately 650 million people that go hungry each year due to fungal pathogens. And now moving on to something that you might not be as aware of is that unlike plant pathogens, human fungal infections have been really rare in the past. It wasn't really until the 1950s when antibiotics, immunosuppressive agents that all revolutionized modern medicine created these conditions where large amounts of bacteria were just indiscriminately killed off in the humans. And it left fungi with new niches to colonize. And so whilst we have medical advances that started to save countless lives, they also enabled this beginning of a shift in the epidemiological landscape of the type of microbial infections that actually pose a threat to us. Now, on your right, you'll see this pie chart that highlights fungal infections and death rates in 2016. And while you might think, oh, mortality looks low, at 0.15%, this diagram is actually a bit misleading, as over 1 billion people are afflicted with fungal pathogens globally. And so 1.5% of 1 billion leaves 1.5 million lives that are actually lost each year due to these fungal pathogenic infections. And really compare this to the 1.8 million lives that were lost to COVID-19 in 2020. I hope that with this kind of description, you can start to see the cause for concern. Now, I really don't have time to go into all of the human fungal pathogens, but there are a few major ones, all that are associated with high mortality rates if the infection becomes severe, that I want to bring up and tell you about. The first set are Aspergillus species. These are actually really common airborne fungi that once they enter your lungs and they're able to overcome your immune cells, become responsible for about 3 million cases of chronic lung disease. And they're also a substantial cause of fungal-associated asthma, which affects 10 million people worldwide. Next, we have Candida species. Now, Candida species tend to be human symbionts. This means that they're microbes that naturally live inside of all of us. However, despite this fact, it's a part of the healthy human body, yes, but it causes infections in our mouths or vaginal regions due to its overgrowths. And also they become really problematic when medical treatments create these environments where Candida albicans proliferates unchecked. We have either the treatment with antibiotics, again, killing off a lot of bacteria, or we have the insertion of catheters where they can grow and form biofilms. And it's these types of infections that lead to death due to the development of intense bloodstream infections. And these bloodstream infections carry the microbe around the body and they cause invasive candidiasis in infected organs. And honestly, it's associated with mortality rates as high as 70% in some countries. With these two, I'm hoping you're starting to see the bad with fungi. Now, the last fungal pathogen that I want to talk about are actually the Cryptococcus species. These microbes that are commonly found in the soil can cause very serious infections that begins with the inhalation of desiccated yeast cells or spores from the environment. And once they've established an infection site in your lungs, they can be disseminated to the central nervous system. This will lead to fungal meningitis as well as bloodstream infections. And what's really interesting to note here is that Cryptococcus species tend to kill a bit more than tuberculosis does each year. Yet you hear and you know far more about tuberculosis and our push to eradicate it than you often hear about fungal pathogens. And what I'm hoping to do here is not really scare you into thinking, oh my goodness, I'm going to die from a fungal infection. But I want to highlight, that fungi in general are it's these groups of organisms that cause very serious diseases, and they're so often neglected. And this is something that all humanity should really be paying more attention to. Now, outside of fungal infections, we also have many animals that face diseases that seem almost apocalyptic to us. And fungal pathogens are really diverse in the ways in which they go about being a pathogen. One of the most common examples is the infection of ants by the Ophidocordyceps fungi. Now, this is an infection that really begins when a fungal spore attaches to and degrades an ant's exoskeleton. Then the fungi begins growing in a yeast state in the bloodstream, and eventually it grows to such a level where it can grow between the ant's muscle cells and then begin to form this dense network of fungal cells that just completely hijack the ant's body. Now, this is some really scary, ugly stuff, but with the ant under the fungus's control, it climbs up off the ground and then really clamps on with its mandible to just like a bean on a huge leaf. And as you can see from the pictures taken, the fungi begins to grow this fruiting body that just like protrudes out of the base of like the ant's or like the tarantula's neck. And it's the height of like the insect that allows for the fungal spores to be carried on the wind and onto like their next target to repeat the process just all over again. Now, a little less horrifying but still extremely concerning, is the infections of amphibians by the Batrachochiritrium, that's a word, fungus. Now, this is a skin infection that leads to the thickening of the amphibian skin, which ends up disrupting the osmotic balance in its body, which is honestly, as you'd imagine, never good. It leads for these amphibians, unfortunately, huge series of cardiac arrests and deaths, affecting animals from frogs, salamanders. And although the disease is less horrifying than the Ophidiocordyceps that I just described, this fungus is actually linked to the increasing decline of amphibian populations globally. And it's currently thought that with this increase in infections and death is caused by climate change. With these increasing global temperatures, these amphibians just end up being more susceptible to these infections, and the geographical range of the fungus is starting to grow. Now, as alarming as all of these examples are, we have some tools in our back pocket that really allow us to treat a lot of these infections for both plants, insects, and animals. Most of us here have heard about antimicrobials, which are compounds that really limit microbes by stopping their growth or killing the cells directly. With fungi, we have antimicrobial compounds called antifungals. And antifungals are amazing because, well, they really help us treat these pathogenic fungal infections I've been describing to you. And what I'm going to do is just walk you through some of the more common classes of antifungals using a handy-dandy fungal cell cartoon. So there are three main classes of antifungals that I want to talk about, and they're all represented by the red squares in this figure. First, we have the azoles. They're antifungals that prevent fungal growth by inhibiting their ability to create new components that are essential for the fungal cell membrane. Without it, the fungal cell can't really continue to grow, and so it just stops fungal growth. Then we have the pauleins. The pauleins are antifungals that really just physically bind to components of the cell membrane. It forms these antifungal-induced pores in the membrane and causes the cell to literally rupture, killing it. And finally, we have the echinocandins. These are a cool group of antifungals that, similar to azoles, they prevent the creation of new cell components. But instead of targeting the cell membrane, they target the cell wall. And without a cell wall, it essentially really weakens or removes the pathogen's ability to survive in its environment. And the echinocandins end up killing the fungal cell. Now, I really wish that this victory was the end of knowing how we deal with fungal pathogens, but unfortunately it's not. And there's something else I would like you to remember, which is that sometimes fungal pathogens can get random changes in their DNA through the process of evolution and natural selection that allows them to really, resist the antifungal's lethal effects. And it gives us a new problem where we know it as antifungal resistance. Now, often fungal pathogens have a highly plastic genome that allows them to generate a huge amount of genetic diversity, helping encourage this resistance. And these resistant fungal pathogens tend to have a lot of mutations that let them resist the traditional drug treatment options. The mutation usually results in either altering the drug target, so they can change it and the drug ends up not being able to have any activity. They can upregulate the expression of the drug target itself. And when you create more targets in the cell, you need essentially a higher concentration of antifungals to achieve the same effect. And lastly, they can just simply export, the drugs out of the cell via efflux pumps. And if there's no antifungal drug in the cell, then there'll be no antifungal effects to be had on any of the pathogens. And so this actually also isn't where our problems stop, because not only do we have issues dealing with the fungal pathogens that we know, but a lot of human activity is actually causing the rise of new and emerging fungal pathogens. And, everyone here knows that we have an incredible amount of data showing how human activity has led to the increased warming of our planet. This creates a huge set of problems, but specifically for this presentation, it impacts fungal pathogens in a really interesting way. This is simply because the ability of most fungi to infect humans is thought to be as a consequence of the way they're adapted to a warmer ecological niche. We often say that fungi tend to be accidental pathogens rather than intentional ones. And the key factor that provides a barrier to infection for warm-blooded mammals specifically is our core temperature at 37 degrees, because most fungi are just simply unable to grow at that temperature. This is known as thermal restriction, and it's really thought to be our greatest weapon against. I think you can see where I'm going here with this, but unfortunately, with the warming of the planet, fungi now also have to adapt to warmer temperatures. And it's this that increases these issues between us, because once they've adapted, they can now infect us with relative ease. Now, for example, in 2009, there was a new species of Candida pathogens called Candida auris, you're seeing in the yellow on the screen. And it was identified globally and thought to originate from the environment. Candida auris, naturally possesses genes that make it pathogenic to mammals. But because of the thermal restriction that used to exist, it never really was a problem for us. And it's currently thought that the increase of global temperatures is what allowed it to be able to now only start infecting us. Now, additionally, remember earlier when I said that agriculture causes some problems for us in the way that we have it set up? Well, one of the ways we ensure that fungal pathogens don't actually destroy all of our crops is the widespread use of antifungals. Now, the widespread use of antifungals do protect a lot of our food sources. And if you'd recall, on my slide that I said just earlier, when it comes to antifungal use, it also leads to antifungal resistance. And so unfortunately, something that helps us also hurts us in another way, because this process of spraying all of our crops with these antifungals to protect them leads to really, really high rates of environmental antifungal resistance. And so something else to know is this Candida auris species I was just telling you about. It's also highly, highly resistant to most forms of the antifungals that we have. And so whilst it was only recently discovered, it's also a pathogen that has a really high mortality rate. And there are cases like this that are starting to pop up and become more common. And we have another example where Aspergillus species that I was telling you about earlier, now people are being found with antifungal resistant Aspergillus species in their body, whilst they've never been exposed to antifungals before. And it's thought that it's this use of antifungals in our environment that contributes to this growing problem. And so please, you should expect to hear more about how emerging fungal pathogens are really coming about and really becoming problematic due to the human specific impact that we have on the environment around us. Now, that's definitely not a note that I'm going to end on, because it wasn't bad or ugly, but straight up menacing. Where I really want to end off is what current scientists like myself are doing to address the threat of fungal pathogens, both old ones and the emerging ones. And it's a bit different than what I've been talking about so far, because it has to do a lot more with the genetics and the physiology of the pathogens. So we do all of this by following something called the central dogma, where molecular biologists believe that DNA makes RNA, which then goes on to make protein. And we can infer the interactions between these three based on the output of the cell that we're studying when one of them are disrupted. In my lab, we do a lot of genetic manipulation at the DNA level using something called CRISPR-Cas9. And in short, Cas9 is a protein that can be guided to precisely target specific DNA sequences, and it mediates these double-stranded breaks in the DNA. We then are able to use CRISPR to add genes that may be of benefit, but we mostly use it to really just knock out genes and study the resulting virulence, pathogenesis, drug resistance, or even really the basic biology of the organism. And once we end up mutating a pathogen how we'd like it, we can then put it in a mouse, so we can put it in a test tube with drugs, and we can record how well it functions, even though we've altered the protein in some way. And in its entirety, we have different ways to study these mutants, and we can look at the genetics, proteins, and even overall metabolism. This sort of combined approach allows us to identify everything from potential biomarkers, and it even allows us to describe the fine-tuned interactions that these microbes would have in regular infection settings. And where we stand right now is amazing, because we can quickly analyze any organism, again, old or new. We can find out what are its potential disease phenotypes, what toxins or hormones that make it pathogenic, or even, what parts of the cell are really essential for its specific survival. And once we understand those things, we can then create new compounds that end up making very good drug targets. And this is the way that you should really be thinking about how people study fungal pathogens. And it's really exciting, again, because it results in us having rapid, specific targeting of these pathogens, and it allows us to effectively treat a lot of the hosts, both human and non-human, with limited detrimental effects to them. So honestly, with that, I just want to say thank you so much for your time and attention today. And if you have any questions, feel free to find a way to reach out to me. Thank you, RCIS, for the opportunity, and I will hopefully see all of you soon. Bye.
So that was such an amazing presentation from Joshua. And as a reminder, if you have any questions specifically for Joshua, that he provided his contact information, so feel free to reach out. So thank you, Joshua, again for that. So we have so many fantastic questions in the chat already, but we had a fantastic question from David about what the other uses are there for fungi besides as a food source. And we're so excited that we have a whole section about this that's coming up. And this is what Jessie is going to be talking about next. So I'd like to introduce Jessie. Jessie McAlpine is a senior PhD student in the Cowan Lab at the University of Toronto. Her research involves studying the interactions between human fungal pathogens and the friendly bacteria living in our bodies. In her spare time, Jessie loves the outdoors and spends most of her time hunting for mushrooms on hikes and trail runs. Thanks, Jessie.
Thanks so much for the introduction, Amanda. Hello, everybody. So I'm super excited to follow up on both JP and Joshua's talks. They gave lots of awesome examples of the many ways that fungi are good and bad. And now I'm going to give some extra examples of why we all love fungi. So I'm going to be talking a little bit about the applications of various fungi, everything from the ones we eat and buy in the grocery store to the ways that they influence medicine and the drugs that we have available to us. So briefly, I'll just touch a little bit on me. So I study microscopic fungi, like the ones that Amanda introduced at the very beginning of the presentation. So I study fungal pathogens that infect humans. And specifically, I try to find new ways to treat these fungal infections by studying bacteria that are found in our body and trying to see if they create molecules that can affect the ability of fungi to cause disease. So I do this at the University of Toronto in the Cowan Lab. In the middle here is a photo of the Cowan Lab prior to COVID when we could all be maskless indoors. And I love my PhD and I love studying fungi. But when I'm not in the lab, usually what I like to do is be out in the forest looking for these examples of macroscopic fungi that, in my case, you can find all across Southern Ontario. And here's a few of the different spots that I've had throughout this summer. But today I want to focus a little bit on some of the applications of fungi. And a lot of what I'm going to be talking about is in this really awesome report that you can check out online if you're interested in learning more, that was done by the Kew Gardens. And it's the State of the World's Fungi report from 2018. And in this report, they cover all kinds of fantastic things, from the applications of fungi to new species of fungi that have been discovered to some of the fungal biology basics that we've talked about today. So I'm going to start off with one of the very obvious ways that fungi impact our lives, and that's the fact that we eat lots of fungi. There's about 350 species that are currently eaten, but scientists predict that this is actually an underestimate, as it can be quite difficult to get a grasp of the entire global consumption of fungi, especially in indigenous communities. And so here I've just highlighted a few of the examples that you might have eaten, either purchased at a grocery store or potentially foraged from around Ontario. So portobello mushrooms and cremini mushrooms are ones you might have bought in a store, along with oyster and enoki mushrooms. Chanterelles, morels, and lion's mane are frequently foraged around Ontario and around the globe as well. And JP gave a really good example of how really the fungi we eat is just a very small percentage of the amount of fungi that are globally available. And so this is only one way that they impact our lives, because fungi also help us make food. So there's fungi that we don't explicitly eat, or I guess we kind of do, usually on the microscopic end, that help us make all kinds of different food groups. So baker's yeast helps us make things like bread and beer and other alcoholic beverages, as well as yeast spreads like Marmite and Vegemite, found in Australia and the UK. Fungi also help us to make hard salami, soy sauce, miso, cheese, and all kinds of foods that you've likely come across in your life and would all not be possible without microscopic fungi that help accelerate the fermentation process to give us these delicious food groups. What's really cool is that fungi also help us make a lot of medicine. About 15% of medicines and vaccines currently available are made with the help of microscopic fungi. And we've also discovered a lot of the medicines that we rely on in the day-to-day from fungal species. So, for example, Amanda touched on penicillin. But there's also other antibiotics that have been discovered as being produced by fungi that can kill bacteria and help us treat bacterial infections. So things like strep throat or ear infections, or the antibiotics that you take after getting surgery to help prevent an infection. Also, fungi are known to produce antifungal compounds as well. So echinocandins are a type of antifungal that's also known to be produced by fungi, and we use those to treat fungal infections. Another really important drug that was discovered to be made by fungi is cyclosporin, and that's an immunosuppressant that has actually enabled organ transplants. So when a patient takes cyclosporin, it can repress their immune system so that they're actually able to take that organ transplant and so that their body doesn't reject it. And without cyclosporin and these other kind of really intense immunosuppressants, we wouldn't be able to have the successful organ transplants that now take place as very common surgeries around the world. You or someone you know may also take something called a statin, which is something that lowers cholesterol. There's many different drug names that fall under that title, but in particular, lovastatin is made by fungi, and that's used very, very commonly around the world as a cholesterol-lowering drug. And there's many other medicines that are either produced by fungi or scientists actually engineer the fungi in order to make. So for example, scientists can take baker's yeast, Saccharomyces cerevisiae, and get it to produce the human insulin protein. And in a lot of cases, that's how insulin is actually produced and then ultimately used by diabetics in order to treat their diabetes. So lots of ways that although fungi can cause disease, they can also help us treat a lot of illnesses. Some of the really cool things I want to touch on today, and we won't have a ton of time. I know we're running out of time a little bit here, but fungi are also now helping us to make sustainable materials. So there's a couple companies out there that are dedicated to basically turning fungi into the everyday products that we need all around us. So there's a company called MycoWorks and one called Ecovative Design, and they're partnering with companies like IKEA and other big companies to kind of modulate how they approach their sustainability goals. So for example, Ecovative Design is making packaging materials that are made out of fungi instead of styrofoam. And that's being used by IKEA at the moment. Fungi are also being incorporated into sustainable decor. You can see down here light fixtures that are made with the similar mycelium instead of being made out of plastic. And then MycoWorks is making things like mycocomposite, which is a type of building material that's being used to replace different structural components. So it can be used to build types of bricks and other types of building materials that can be used for sustainable development. And then one area that I find really cool, and I'll touch on briefly, is that fungi are also helping us to make leather and other sustainable fabrics, where companies, again, like MycoWorks, are turning fungi into a type of leather that's obviously not derived from animals. And so this is a vegan leather that's also sustainable to produce, can come in a variety of colors and textures, and is now being used by companies as diverse as Hermes and Adidas, who have come up with a concept shoe that uses this fungal leather and is not currently commercially available, but should be in the future, and something that will be very exciting to follow along with. So that's my very brief overview of some of the applications of fungi. I want to leave lots of time for the Q&A and to discuss a little bit about foraging for mushrooms. So please send me in lots of your questions, and we'll be able to answer them shortly.
Awesome. Thank you so much for that, Jessie. And now we are going to move on to a section called Some Questionable News. And as a reminder, keep continuing to put your questions in the chat. After Some Questionable News, we will go on and do a Q&A, and we'll get some of those questions that you have answered. And so for this section, this is kind of a relatively new section to RCI Science events, where we present different headlines and our panelists have to guess whether they are real or whether they're fake. And important to note, we're not talking about whether the science is real or fake. We're talking about whether the headline was real or fake and existed or was submitted by a news source. So with that, we also like to encourage the audience to play along. So there should be a poll going on. A new function we're just trying out is a poll through YouTube, or you can also submit your answers in the chat and try to guess whether these are real or fake. So our first headline is, like a pack of teenagers, Manitoba mushroom explosion fueled by amorous fungi, experts say. Is that a real or fake headline? JP or Jessie, anyone brave enough to take a guess?
I'm going to say real in that I don't think they mean explosion like fire explosion. I think they mean like a lot of growth of fungi. So I'll say real.
Yeah, I would say real as well. You could have some of the spore discharges are very explosion like the puffballs. If you step on a puffball, it's big explosion, and the height and the range that they can disperse is much bigger. If you take the size into proportion, that's a real explosion.
Well, great job. The answer is it is real. The audience had a 65% thought they were fake. So good to know. I definitely thought this one was fake, but it is about how this year there were heavier rainfall in Manitoba, and this encouraged more mushroom sprouting. And like JP said, these produce spores that disperse and reproduce and can cause that kind of sensation of explosion. So let's try ourselves with the second one. This headline is, Fighting Fungi with Fungi. Treating athlete's foot with competing fungus shows promise as topical treatment. What do you think?
I definitely think it's real as someone who looks for small molecules secreted by microbes that grow along with other microbes.
JP, what do you think?
It's a promise, shows promise. That's true. But I don't think, as far as I know, probiotic treatment has been used for treating athlete's foot. So
that is, so this one is actually fake, so well done. 76% of the audience thought it was real. I definitely also thought it was real. It sounded like the type of thing that would be cool. And as we know, like Jessie said, we often use these byproducts of fungi to treat different infections.
Was I right?
Yeah, you were right. Congratulations. And congratulations to everybody at home who was right too. Okay, so on to our last one. Scientists say astronauts should take psychedelic mushrooms in space. Real or fake?
Should.
It would be an interesting experiment, so I'll say real, depending on who the scientist is.
I think it's fake because we really don't have a good clinical data yet for any of the psychedelic mushrooms. We have them treating certain mental diseases, but not for, say, what the symptoms astronauts would experience. So I think it's fake.
Okay, and the audience, according to the poll, 52% of the audience believe it's fake as well. But I have to say, this is real. This was a real headline, not necessarily real data, but this was an idea from Paul Stamets that suggested that using psilocybin, an active ingredient in psychedelic mushrooms, could help with mental health of astronauts. And I agree. There's a could, not the should. But it was also part of a larger discussion, just like Jessie had mentioned, about how mushrooms could be used in space to build different materials and things like that. So Well done to everybody who got some of those answers right, because that was really tricky. A good reminder that sometimes just because it is a headline doesn't mean it's real. And I know that JP, you've been entangled in some questionable news in the past. Can you tell the story behind your research on the Midsummer's Nightmare?
Sure. So as I mentioned in the presentation, that it took a long time for the Chinese Center for Disease Controls from the local to provincial to national level to try to figure out the cause. And they could not solve it. And finally, they invited epidemiologists from Europe and from the US CDC. And what they found was the people who died recently had remains of that mushroom, little white mushroom. So I thought it was interesting. Actually, when this happened, the first time I encountered that issue was we were traveling in southwestern China with my host in the Chinese Academy of Sciences and 2 German mycologists. We were collecting mushrooms. And right in the middle of our trip, we were pulled aside to the local Center for Disease Controls to ask us to identify a mushroom that they found. And none of us could identify what that mushroom was. At that time, that was 2006. We did not know. And they have had the sudden unexplained death for 3 decades at least recorded. So we couldn't find. And with a couple of years later, they involved this international team, and the national team suggests that this might be the cause. This is mushroom, and it suggested 2 potential causes associated with this mushroom. One is that it might have a high barium concentration. So barium is a metal, and barium in certain forms, like barium chloride, has been found associated with sudden cardiac arrest. So they suggest this might be a cause that this mushroom might be able to potentially concentrate barium from the environment, and then if you eat it, you can get you cause sudden cardiac arrest. The second one was about toxins, but people have been looking at the toxins for 3 years by then, and they were not able to identify the specific toxin. So I went with the idea that we should be able to look at the barium concentration. And we traveled to quite a few places. In one of the trips, we took a whole day, walked about 20 kilometers in very rainy and muddy condition. And by the time we got to the spot, it was very slippery, and we collected a mushroom, 37 mushrooms. So I think we were saying that probably the number of kilometers equals the number of mushrooms. We collected about 37. And we brought them back. And meanwhile, my daughter and the other team members were staying behind because the road was so muddy and they could not drive through. They found a bunch as well. And we then took those mushrooms and analyzed the barium concentration. And we found that the barium was not the cause. So we wrote this paper up, and it was published in Applied and Environmental Microbiology, and it generated quite a lot of news. So they including CBC and the Hamilton Spectator. So we said, barium is not a cause of the death from this mushroom, but instead what they reported was that scientists debunked the news that the mushroom was the cause of the sudden death. So, I was explaining when I was talking with them very clearly. They said, no, we did not exclude this mushroom as the cause. We exclude the barium in the mushroom as the cause.
Yeah, so interesting how even, when talking to journalists, even how those messages sometimes get misconstrued. Great. Thank you for sharing that story of how these questionable news kind of happen. So next we have, I know we're over time, but we're going to keep going. And if anybody needs to leave, you can watch the rest of the video later. But next we have some questionable news TikTok edition, which is a very, very new session where we are going to have our expert panelists watch a fungi-related TikTok and then guess if the video is true or false. And we'd love the audience to play along as well. So we're going to watch this quick TikTok.
Hey guys, it's me again. I'm in Hawaii, and normally we listen to plants, but a lot of you have been asking, what do mushrooms sound like? And I found this really cool piece, it's a tree in this bamboo forest, and there are all these amazing little mushrooms on it. In fact, I even have my plant wave sitting on a mushroom there, which is nice. So let's check this out. Let's have a listen. We have one connected there, and we'll listen to these connected through the tree.
So obviously we weren't making a fake TikTok. So the video itself is real, but the question we have for our experts is this actually how fungi communicate?
I'm going to say no.
No. I'm going to say no too.
Perfect.
Very relaxing sound, but
exactly. And the audience was able to detect that too. 75% said it was fake. So this is actually a technology called plant wave, where they detect slight electrical variations in a plant via 2 electrodes placed on leaves, or in this case, fungi, and the variations are graphed as a wave, which is then translated into music. So it's kind of this artificial idea of this is how fungi are communicating. But it leads to a great question that one of the listeners came up with, which is a system called the Wood Wide Web. Mycorrhizal fungi form a symbiotic relationship with tree roots that allows trees to warn each other about dangers to them, pests, drought, etc. How does this work? And similarly, how can fungi communicate, and if so, how?
Jess, you want to go first, or you want me to?
You go first, JP. You got this.
So for trees, the majority of them have ectomycorrhizal fungi and endomycorrhizal fungi. The ectomycorrhizal fungi are those that invade the plant root cells, whereas the endo, they invade the intercellular space of the plant roots. And those fungi have mycelium phase. They grow independent of the plant roots. So the same fungus, when they grow out and spread on the forest floor, they can infect multiple trees and sometimes multiple species in the forest. So they are very excellent transport channels for nutrients and water. They can act as a channel for transporting nutrients from one tree to the other, like including the parent tree and their offspring seedling to help the seedling grow. That's one of the mechanisms that they can help each other. And they can sense the stress that the host plant is suffering based on the nutrient that they are synthesizing, the water level, etc. So they can act as a buffer to adjust the nutrient, water, mineral, etc. in the forest. So it's a very good term. Thank you for asking that question. The wood wide web in the forest.
Awesome. Yes, I think that's a perfect way to debunk that they are communicating, but not in the general way that you think they're communicating. They're communicating by sending water and different kinds of small molecules and things like that.
Yeah.
Great. So again, the audience, continue to feel free to submit some questions. We're going to take some of the questions that people have submitted already and present them to our panel. At the end, we will also have a section called Fungi Fun Cam, where we'll show some mushroom pictures submitted by our audience, and we'll have our experts try to guess what species they are. But first, we'll start with some questions. This one I'll give to JP. How do we estimate how many fungal species there are, and how do we know how many that we haven't found yet?
Yeah, that's a very good question. So for a long time, scientists have been trying to guess. And one of the first guesses was based on the ratio of fungi in a forest, or in a few forests in England, relative to the tree number. So England had about a couple 100,000 trees. And the fungi that they were able to identify over there in those small forests was about 1-to-6 ratio. So they think that every tree that they find will probably correspond to 6 different fungi. So they said, okay, at that time, well, even now, the global tree species is about 400,000. So they estimated that probably it's about 1.5 million. So that's what's the earliest estimate, is the ratio of plants to fungi. But more recently, we have more data from global surveys instead of just England. And we have new techniques called metagenomics that allow you to directly sequence what's in the different environments. And there have been a lot of metagenome sequencing, although most of them are for bacteria, but many of them also have fungal data based on specific sequence called internal transcribed space region. And based on the diversity of sequences, unique sequences that you find in the different environments, then you can estimate the number that are likely present in the world that based on that sequence. And depending on the assumptions, so which environment you have sampled, how representative they are, and how the species concept you use to define how much ITS sequence difference you're going to use to define species, then the range can differ very broadly. So as I mentioned, there's 700,000 to over 5 million, and some of them even higher number. And that's, yeah. So just want to bring up very quickly that there's a lot of debates about what should constitute a fungal species. And there's no consensus among fungal biologists. So what should be a fungal species?
Exactly. I was thinking about that too when I think about the Great Potato Famine and how that was caused by oomycetes, which are not considered to be fungi, but they used to be. And I remember when I learned that, I was like so surprised because I'd always thought it was fungi. So there's these concepts that definitions change and debates in the community and things like that change all the time.
Or slime molds. When you find slime molds in the environment, they look like fungi a lot of the time. So even I get confused sometimes and then double check that it's a slime mold.
For sure. I have a question for Jesse from the audience. How are fungi being used to turn crop waste into bioethanol?
Yeah, that's a great question. So this is also covered in the State of the World's Fungi, fantastic report. So I think kind of the traditional way is that yeast is used to ferment. In the past, it was actual crops, so things like maize and corn, ferment it into ethanol that can then be added as a fuel source, as a biofuel. But now they're working on like the second generation of this process, where now they can take basically waste from agriculture. So things like stalks and straw and other parts that we don't consume, and ferment that into the ethanol to be used as a biofuel. And that's hopefully going to be a more sustainable biofuel, since we're not taking crops we would normally be eating and kind of looking at agricultural waste instead to be turned into biofuel.
Awesome. And another question from the audience, what about lichen? Are fungi involved in lichen? And if so, how?
Do you want to answer, JP? Or I can? I don't care.
No, you go ahead.
Okay, well, I mean, I probably know less than you, but yes, lichen is a symbiotic relationship between fungi and cyanobacteria or algae. And so basically the algae kind of live in these filaments that are produced by the fungus, and then they create these very interesting and diverse like macro structures that we can see, but it's a symbiotic relationship between two microscopic fungi. Or fungi and algae.
Anything else you want to add to that, JP?
Sure, yeah. Lichen is a very interesting case in that they are quite often the pioneer organism, pioneer symbionts in many of the novel barren ecological niches, like in deserts, in the Arctic, and even on some of the trees. They would develop and hanging on the trees. So they are very interesting organisms.
Great. Another question from the audience: are any of the fungi mentioned today, do they have psychedelic effects? And that might be a good question for JP because you showed the most different kinds of mushrooms.
Yeah, so the ones that I showed on my slides, none of them are known to have psychedelic effects, but they are some of the ones with psychedelic effects, which there are about 200 of them, 200 species, and distributed all over the world. Some of them do look like the ones that I showed, but not the ones that I showed.
Great. And one of the fun facts on the psychedelic discussion is relates to a question that somebody else had about how the zombie fungi affect the insects. And new studies are showing that the fungi actually prepare or make different kinds of small molecules that then can affect the insects. And in some cases, they have found different psychedelic molecules that the fungi are making inside those insects, and that could be one of the ways that it's controlling the insects and turning them into zombies, which I think is really cool.
I should add, if I may, that some of the fungi are very expensive. The mushrooms are very expensive, like the truffles, the morels, the matsutake, etc. They are expensive because of the cultural value. Quite often, it's not because of their nutritional value. For example, Japanese like to have, buy one mushroom, they would cut one slice for one meal for the whole family to share. It's just a cultural tradition. And the same for truffle in Europe, the black truffle or the white truffle. And that is the smell that the sensation that you get. Whether that you consider that as a psychedelic or not, I guess it's different. But I think for people who are eating them, I think they really enjoy them, give them pleasure.
For sure. And I'm definitely one of those people who loves the taste of mushrooms. I know that lots of people don't, but they provide me lots of joy. Another question: several types of fungi have been discovered that break down plastics. How can these be used to clean up landfills and the ocean, and which ones are best suited to that task?
Jesse?
Yeah, I was going to say, I don't know if I know of any specific types, but one of kind of how a lot of fungi grow is that they secrete enzymes into their environment that can break down different components, and then they reabsorb those nutrients. And so that's kind of the basic process of how fungi eat. And so a lot of scientists and research has gone into discovering whether or not fungi can break down things like oil, plastic, garbage. And yeah, as far as I know, there are mushrooms that are being used for like remediation of oil spills and for breaking down of plastic. But I actually don't know of any specific species off the top of my head. I don't know if JP does.
So those plastics or artificial chemicals, what they call the xenobiotics, quite often they are long polymers and very stable. They have those phenolic chains with chloride and other kind of chemicals associated with them to enhance the stability. And that's one of the reasons why we use them. So because of the stability, most of the enzymes that we have in the environment cannot really break them down because it takes a lot of energy to break those phenolic rings. But fungi, there are several groups of fungi that are known to be very good at using stable compounds as the energy and the carbon source. So the white rod and brown rod, for example, those fungi, they have a very active oxidizing enzymes that can add oxygen or hydroxide to those rings. And once you start adding those oxygen or hydroxide to the rings, they become less stable. And then the enzymes can come in and cut them, break them apart. And that will subsequently be used by the fungi as carbon or energy source. So they have those kind of enzymes. And we know quite a lot of fungi have enzymes that can break down chitin. For example, many of the wood decaying fungi, they would have those enzymes.
And this brings up perfectly a discussion we had last year in one of our events about using mushrooms to create coffins, and so that you can have the decomposition of the human body, and really harnessing the power of the fungi. That's great. Okay. Another question we have is, I think this was related to Joshua's presentation. Joshua mentioned that global climate change has affected the geographic range of various fungi. Somebody has asked, how does that happen?
It's a very difficult question. It's a very good question. And I think a difficult question to really answer it very well. But we can glean from the data. So we know that at least from the macrofungi, mushrooms, they're very dependent. The fruiting is very dependent on the climate. For example, why do we see mushrooms in the fall? Because the temperature is now low and there's a lot of moisture. When we have those conditions, we see a lot of mushrooms. If it's dry or high temperature, they typically do not fruit. So using those conditions, those kind of indicators for fruiting, then we can see how the climate change could potentially impact mushrooms at least on the micro scale level. So if we have more severe droughts, hotter weather, we're not going to see mushroom fruit as much, and the growth probably will also be impacted. And another angle to look at it is through studies of metagenomics, doing global analysis. So there's been a report of metagenomic analysis of global samples, try to identify the fungal diversity, the relationship between fungal diversity and the climate and the soil physiological characteristics. And one of the best indicators is moisture. So the higher the moisture, the more diverse group of fungi you will find. And then we found this in our own survey of the global yeast diversity that we just published in iScience a couple of months ago, where we found that moisture was associated with yeast diversity at the global level. So when that distribution changes, the moisture level changes, we're going to see the changes in fungal diversity. And the third level to look at it, the third angle to look at it, is through analyzing the altitudinal distribution of fungi along, in a mountain, in a big mountain. So southwestern China, that I have been doing some of my field work, my collaborators have been looking at this distribution from as low as about 200 meters above sea level to like 4,000 or 5,000 meters above sea level, all in one mountain. And you can do this kind of survey to look at year over year how the fungal diversity has changed. Then you can gain a little understanding of how the warming trends would impact those. And we have seen some of those evidence of changing distributions of the mushrooms on those gradients, altitudinal gradients.
Great. And, my PhD research was about how temperature affects the traits of fungi as well. And we find with fungal pathogens, many of them, elevated temperature actually affects its virulence traits and enables them to be able to cause disease through those virulence traits. So there's lots of ways that temperature can change these fungi, as well as, our body is 37 degrees because it's able to, one of the things about our body being 37 degrees is it's able to kill off a lot of bacteria and fungi who aren't able to grow at those temperatures. But the growing temperatures of the earth is allowing more of those fungi to be able to adapt to those increasing environmental temperatures, which could mean that there will be more fungal pathogens that will be able to survive within our bodies as well, and therefore cause more disease. So there's lots of different ways where climate change could have an effect on fungi. Yeah,
I just attach along because Joshua is not here. he talks about the Candida auris. So one of the hypotheses about the sudden emergence of multiple clades. So Candida auris is divided into 5 clades, and within each clade is very clonal. They are all very genetically very similar to each other. So the reason why there's a reason for sudden emergence of multiple clones of this species in the hospital environment, and causing very high mortality rates. So earlier this year, my colleagues and I, in our group, identified for the first time of environmental reservoir for Candida auris. And that was in a very pristine environment in Andaman Island in the Bay of Bengal, which is very traveled by people. Over there, we found Candida auris. And we also found a multi-drug susceptible strain, as well as multiple drug resistant strain. And the drug resistant strain was genetically very similar, only different at the whole genome level by 1 nucleotide, to a strain that they found in the hospital in India, in the mainland India. And this definitely lends support to this global warming hypothesis in that the fungus are in the environment. They are in the marine environment. They have to have many of the stress responses in the environment to deal with those environment. And that same kind of stress allow them to survive in hospital environments, including some of the disinfectants that will apply to them and some of the drugs. So they may share very, very similar pathways to the drugs that we use to treat, to kill them.
Yeah, absolutely. So with that, we'll wrap up the question session. Thank you to everybody who submitted questions. And if your question didn't get answered, we are so sorry that we ran out of time, but please feel free. I know I'm always happy to, if you reach out on Twitter or LinkedIn, I'm always happy to answer questions. I'm sure other people on the panel would be as well. So feel free to keep that conversation going. With that, we'd love to switch to our last session, which is the Fungi fan cam. We are so lucky that we have some pictures here of different fungi, and we want to ask our experts if they can identify the species. Now, we don't actually have an answer because you're the experts, and so we'll take your word or we'll take your advice on this. And we'd also just love to know what you think more about this. So we'll get started. Any ideas about what this fungi is? I will say, as I'm a mycologist who knows nothing about macro fungi, I only know about the micro ones, so I will not participate, and I'll leave it to the two of you.
I think this looks like a turkey tail, Chamaetis versicolor. It's very common in Southern Ontario. But
that would have been my guess, but it's a little hard to tell. You often find them growing on dead logs, dead stumps, and very common in Southern Ontario. If you Google turkey tail, you can see like different stages of maturity, and they can be really cool and kind of colorful as they get larger.
Great. Okay, and we'll move to the next one. The location of where they was found or on the bottom.
This is good. We've got one. Okay, so I'm not going to know the exact scientific name. Maybe JP will, but this is a coral, which is one of my favorite types of fungus. Also pretty common in Ontario as well, and throughout Canada. There's different types of coral, and they're named like that because they obviously look similar to coral that you might find the sponge that grows in the ocean. But JP, do you know?
Yeah, coral fungi are largely, most of them grouped into one genera called the Ramaria. And the classification within that group is really not very well defined. But it's the company called Ramaria. It's
awesome.
Coral mushroom, yes.
And a reminder that the audience is welcome to play along too. We want to hear your guesses as well. Okay, this next one.
That's a good one. This is Russula mimosa. If it's found in North America or Europe. If it's found in Southeast Asia, it's called a Russula canosa, because I worked on this mushroom. It's very bright red. There's actually a story about this in Southeastern Asia and Southern China. This mushroom is harvested, dried, and sold as a gift to people who have just recently given birth. And the superstition is that the red color symbolizes the blood, in that when you give birth, quite often they lose a lot of blood. And by giving the woman, the mother, this mushroom, the help, the thinking was that it could help them replenish their blood because of the red color.
So what do you think about this one?
Bear's tooth, maybe?
Hericium?
Yeah, okay.
Yeah. We call that monkey head mushroom with all the messy hair.
Yeah. Or I think bear's tooth might be another name for it.
Mm-hmm.
This one looks like something decaying. That's all I can. It's a beautiful color.
Yeah, it's cool. I actually don't know. Someone has said hedgehog fungus. Okay, I don't know.
Yeah, I think it is. Some of the turkey tail also look like this as well in certain stage. And we can't really see any of the underside.
Yeah, it's hard to see.
Yeah. If the underside has a lot of hanging threads-like structure, and it looks like a hedgehog, that would be it. Capranus comatus.
Yeah, this is what I hoped we'd get. Shaggy mane, inky caps. These are very common, and you see them often on lawns, even just walking in the suburbs.
Yeah,
I definitely have this one growing on my front lawn.
Yeah, there's a bunch in the backyard, and my dog always looks at them suspiciously.
Yeah, Boletus. Difficult to say which species. I think it's Boletus. So Boletus is a very large genus, hundreds of species. My collaborator and I recently we published like a 220-some page paper in Fungal Diversity, describing and sorting out all the species in the family Boletaceae. But still, there's a lot that we don't know, and many of them cannot be recognized based on the morphology alone. Have to use DNA sequences or microscopic structure, microscopic observation to know the name. I think
there's definitely some sort of Amanita on the right-hand side.
Yeah. The right side is Amanita muscaria. The left, I think, looks like a turkey tail.
Yeah.
Chamaetes versicolor.
We're really having a hard time stumping our experts. You are both doing so well.
Having a hard time stumping JP.
No. So I'm not a fungal taxonomist, as I mentioned earlier. I know the common ones, but sometimes the most difficult ones are the ones that look like it has no really distinguishing feature. Many of these ones over here have quite a distinct feature.
Yeah. I think that's another Hericium on the left-hand side. So something like bear's tooth or, monkey head. Mm-hmm. And I don't know, honey mushroom, maybe?
Yes, you're right.
Oh,
I think that's what Amanda, I think so. I think that's the one Amanda mentioned, the humongous fungus among us.
Yes. Also pretty common, yeah.
So on the right, I think it's puffball.
Yeah.
Right? And maybe not.
Oh, but you can see a stipe, like a stalk.
So if there's a stipe, it's not puffball. So this is, I think, Chlorophyllum lepidites is a poisonous one on the right. In the middle, it's really hard to see. Could it be?
It's also hard to tell if it's purple or not. If it's
right. Could it be a Lactarius?
Oh, like a milky cat?
Yeah.
Maybe.
If if you break it and have a milky substance, that could be.
Those are my favorite when you see the videos. For anybody who likes looking at mushrooms, please explore Fungi Twitter and Fungi Instagram, because there are so many great accounts that show really good ones. And I love seeing the ones, but when you cut them open, it looks like they're bleeding or that they're dripping.
Latex.
Yeah, there's so many cool different kinds. So I think this is a great way to start and say, I think from all of this talk about mushrooms and how pretty they are and all of the cool things that they do, many people might be into and getting excited to try foraging. Do you experts have any advice for people who are looking to get started in foraging, what they should look out for, and any tips?
Yeah, I have 2 slides that we can touch on, and then maybe JP can add to those if we look at my screen again. So I know JP touched on this briefly, but I just wanted to reiterate that foraging can be a lot of fun, but it can also be very dangerous if you're not 100% certain of what you have identified, or if you're not with an expert, it can end, unfortunately, very poorly. In Ontario, we don't often think of dangerous, toxic species. We often think that that's in kind of lands abroad, but even commonly in Ontario, we can find on the left-hand side here the destroying angel, this species of Amanita bisporigera. And this is commonly mistaken for species such as the horse mushroom or the meadow mushroom, especially with people who are just beginning to identify and forage mushrooms. And kind of one of the general rules is that if you see a white mushroom that has white gills and a veil, this sort of veil or skirt that you see, definitely leave it alone. Those are very, very toxic mushrooms that are quite common, at least in terms of Ontario. And so I outlined just a few of the etiquettes that I sort of follow when looking at foraging. I know there's a lot of text here, but kind of the main takeaways are that if you're not 100% certain that a mushroom is edible, just sort of leave it alone and appreciate it. It's something that you have come across, and definitely keep in mind kind of local laws and landowners. Private land and trespassing is not good when it comes to foraging for mushrooms. Unfortunately, another common etiquette is that a lot of mushroom foragers don't necessarily want to be asked where their special spot is located. People can kind of bristle at this, so sort of respect that very experienced mushroom foragers may not be willing to give up their prime locations. Some of my common kind of tips are to bring a small knife with you. Depending on who you're with, cutting mushrooms can help you with identifying them. But it can also help you determine whether or not the inside might be rotten or past its prime and not ready to be eaten. It's also really, really commonly recommended that even if you're 100% certain that a mushroom is edible and you're going to eat it, start by cutting, cooking, and eating a very small amount, because you can still be allergic to mushrooms, even if they are edible. Even if you've eaten, kind of your standard grocery store mushrooms, you can still have an allergic reaction to mushrooms that have been foraged. Also, a rule that I follow, but this is not a strict rule, is that if you find a patch of mushrooms, never take more than about 25% of what you can see. This ensures that you don't, destroy an entire patch of mushrooms, and also leave some for any other foragers who might be looking. And of course, if you're uncertain, just leave the mushroom alone. I always find it can be just as fun to spot fungi and kind of guess at what they might be, but not necessarily eat what you found. And I'll let JP add anything that I may have missed there.
No, I think those are very good recommendations. Yes, I don't have any more to add.
Okay, so thank you so much, both of you, and as well as Joshua, for that. All of the amazing insights you provided today. And the take home, everyone, from today is be careful. Mushrooms are great, but they can also be very dangerous. So if you want to get it started with foraging, please make sure that you're cautious, because there have been an increase in poison control has dealt with a lot of people trying to eat mushrooms that were not edible. So definitely be safe.
Yeah, and that applies to your pets as well. There was a recent Hamilton case of a dog eating a poisonous mushroom and died.
Okay, yeah, so watch when you're taking your pets on walks as well. So thank you so much, Jesse and JP, for joining us today. We learned so much. I learned so much from both of you as well. And to Joshua, for even though he couldn't make it and even though he's sick, for still sending a video. We're so grateful for all of you. And with that, I'll pass it back to Suzanne.
Well, thank you to all our speakers, especially Amanda, who chaired the whole thing. Thank you. We hope you enjoyed today's discussion as much as we did. I certainly learned a ton. And what fantastic questions from our obvious fungi fans in the audience. I thought that was incredible. I've never seen such good questions. Please share your feedback with us and let us know what you'd like to see from us in the future. There's a link in the chat box. And if you can, consider supporting RCI Science's free programming. You can visit RCI Science/donate for more information. Now, November, next month, is Diabetes Awareness Month, and you can join us as we continue to celebrate the 100th anniversary of the discovery of insulin. We have a weekly series of short conversations with author and podcast host Krista Lamb and diabetes researchers working at the intersection of science and society. And also follow along on Instagram we have wonderful takeovers from scientists across Canada. You can get all of our upcoming events on our website, rciscience.ca/what'son. Again, link on the screen. And thanks to each of you for joining us. And again, thanks to our terrific speakers. Don't forget to like and subscribe on social media. Bye.